Welcome to Bachem, the market leader in the production of peptides and a specialist in the process, development, and manufacturing of active pharmaceutical ingredients. Our GMP manufacturing sites are located in the USA, Torrance, California, and in Switzerland, Vienna and Bubendorf, where the headquarters are located. Bachem has a quality management system that covers the entire process, from incoming materials to the production, release, packaging, and delivery of the final product. For the large-scale synthesis of peptides, we require not only amino acid derivatives, our building blocks, but also reagents, solvents, and other chemicals for performing the chemical reactions. Our logistics system guarantees the timely supply and storage of the required raw materials. Every year, thousands of liters of solvents are required for the large-scale synthesis of peptides and their purification by preparative chromatography. For storing and handling such large amounts of various solvents, we are equipped with a state-of-the-art, fully automated tank farm with underground tanks. Peptides are short chains of varying amino acids connected by so-called peptide or amide bonds, which involve the carboxyl group of one amino acid and the amino group of the other amino acid. To obtain a peptide with a defined structure, the amino and carboxyl groups, which should not react, have to be reversibly protected. After formation of the peptide bond, the protective groups have to be removed. Our amino acid derivatives are either produced in-house or purchased from reliable manufacturers. All chemicals required for the production of peptides must be thoroughly analyzed and released by our quality control department before use. At Bachem, quality matters at each step of the synthesis, from any raw material to the final product. Bachem's policy is to keep all required material on stock so that orders can be fulfilled rapidly. Bachem's peptides are synthesized by chemical methods, either by solid phase synthesis or by synthesis in solution. The choice depends on the structure of the peptide, customer specifications, and other factors. During solid phase synthesis, the growing peptide is linked to an insoluble carrier. The synthetic steps are monitored by taking samples for in-process controls. With more than 20 reactors, the largest one with a volume of 1,000 liters, Bachem has the required equipment for producing kilograms of peptide. The rapid method lends itself to automation. Small-scale syntheses are run fully automated in state-of-the-art synthesizers. When performing GMP syntheses at kilogram scale, only the washing steps are software-driven, whereas the critical steps are monitored and documented by our experienced specialists. Eventually, the peptide has to be split from the carrier resin. During this step, the peptide is completely deprotected. The resin is filtered off and the crude peptide is isolated by precipitation. Several kilograms of material can be obtained using reactors with a capacity of up to 1,000 liters. The removal of the solvent from the precipitate can be rapidly and efficiently achieved at large scale by inverting filter centrifugation, or for smaller projects by standard filtration. The crude peptide is then dried under vacuum in a rotary evaporator or in a drying oven to remove any remaining solvents. When synthesizing peptides in solution, smaller fragments of the desired sequence are prepared first. They are eventually assembled to yield the complete peptide. Unlike the solid phase method, the intermediates can be isolated, characterized, and purified. Bachem can produce hundreds of kilograms of peptides using solution phase reactors of up to 8,000 liters of volume. Peptides obtained by solution synthesis are purified either by crystallization or by countercurrent distribution. as the desired product and its impurities can differ in their solubility in water or organic solvents. Countercurrent distribution is equivalent to a continuous extraction, achieved by repetitive mixing and separation of two liquid phases. The method is fully automated with extraction volumes of up to 400 liters. Preparative HPLC is the standard method for purifying peptides obtained by solid-phase synthesis. A solution of the crude peptide is applied to a steel column filled with silica-based material. Our large columns with a diameter of up to 60 centimeters allow the purification of up to kilograms of crude peptide in a single run. Large-scale preparative HPLC requires a vast amount of eluents. Up to 2,000 liters of eluents can be prepared in our large tanks. In-process controls of the obtained fractions are performed by the Quality Control Department. Here you can see a typical chromatogram of the fraction containing the purified peptide. Lyophilization or freeze drying is a gentle process for obtaining the purified peptide in solid form. Peptides used as pharmaceutical ingredients have to be lyophilized in clean rooms. Our lyophilizers allow us to isolate several kilograms of peptide in a single run. Bachem has clean rooms of all required classes for isolating and handling of APIs. Bachem has 40 years' experience in producing peptide APIs. We are totally committed to full GMP compliance, which has been acknowledged by the FDA and other national authorities who inspect our facilities regularly. Our consistent focus on and appreciation of our customers has allowed Bachem to establish itself as the market leader in the manufacture of peptide APIs. Bachem, quality matters.